I told you that phytoalexins are secondary metabolites which are not synthesized all the time, but they are species specific, pathogen specific response. Now this involves therefore de novo synthesis of the enzymes, which includes transcription, translation, all the process starts after the pathogen has attacked. So these phytoalexins are a very wide range of secondary metabolites, sometimes which are very broad range pertaining to a particular taxa or taxonomy, or which are very specific to a particular kind of insect which is, or the pathogen which is continuously attacking that particular species. So they remain generally undetectable in the plant before the infection. So now what other mechanism by which the plant tries to protect itself? You remember I was talking about acquired immunity and innate immunity in plants. So we will be today talking about the different acquired and innate immunities in plants. Now some plants, they recognize specific pathogen derived substances. They can recognize particular antigen epitope of a particular broad taxa, like for example bacteria which are movable, so they will have flagella. So there are epitopes present or the receptor present on the plant cells which can recognize motile bacteria. So by this they are able to have a generalized defense against any such bacteria which is motile in nature, which comes and attacks the plant. Now through evolution they recognize these specific components of their pathogens, through which they then develop the specific antibodies which can recognize the antigens, and then which can further trigger the secondary metabolism specific to that particular taxa or that species. Now individual plants, we know that they differ in their immune system, their sensitivity or their capability to resist the attack. That depends on two things. One is the array of secondary metabolites, and the second is how quickly they recognize and induce the secondary metabolism. Now the resistance depends on how plants sense the presence of pathogens and initiate defense responses. Now the first line of resistance is therefore provided by a system that can recognize broad categories of these pathogens. Now these are called as microbe associated general molecular patterns, MAPs. Now these elicitors are evolutionary conserved pathogen derived molecules, such as basically first some structural component will come into contact, their cell walls. So therefore you will see that through evolution with their contact they learn the plant. So there is a memory in the plant through which it is learning and creating its own defense for future. So structural elements for fungal cell wall, for example, or the bacterial flagellum. So one such is the flagellin receptor FLS2, which enables the plant to recognize all mobile bacteria. That is one such receptor. Then another, as I said, is called PEP13E, which is an epitope for omicide transglutaminase. Now it recognizes transglutaminase and then it will induce its defense response. So this can recognize broad range of omicides. So this form of defense strategy comes under innate immunity, which through evolution the plant has learned, it is there within. Now when a plant survives first infection, then it can resist. It has been observed that at its subsequent resistance increases. So their first infection is said to improve its resistance, so they are able to show better resistance during further infections against the similar kind. Now this phenomena is called as systemic acquired resistance. Now what is involved in systemic acquired resistance? It develops over several days. It is not that suddenly after the infection. The plant develops after days from the first point of infection. How does it happen? Now there are increased levels of certain PR proteins like chitinases and other hydrolytic enzymes in the plants. in the process of systemic acquired resistance. Now, one of the endogenous signals involved is likely to be salicylic acid. You remember salicylic acid was one of the molecules involved in the signal transduction pathway. So, now salicylic acid and its volatile components like methyl salicylate, they are said to be responsible or involved in this systemic acquired resistance. So, there is a phloem induced signal. So, once the pathogen attacks, now these are involved which through the phloem signal to other parts of the plant, and because methyl salicylate is a volatile component of it, so the nearing plants also would therefore induce the systemic acquired resistance. By inducing what? By inducing the production of PR proteins. Now, another such signal is H2O2. Now, H2O2 is another compound that accumulates at the site of infection and plays a role in SAR. So, this is how it is also linked to, I was talking about reactive oxygen species. So, at the point of infection, so multiple, it is not that only one defense will be playing a role, but there are multiple modes of defense which come together to protect the plant. So, it is that hypersensitive response that the same reactive oxygen species or H2O2 which is generated is then helping in the SAR also. Like salicylic acid, H2O2 is unlikely to function as a long distance signaling molecule. So, you need volatile components. So, this is a picture which can demonstrate the systemic acquired resistance. So, upon infection, from the site of infection, then it induces a phloem signal which further accelerates the production of salicylic acid or methyl salicylate, and then it further accelerates or induces the production of, elicits the production of PR proteins. What are PR proteins? Pathogen related proteins. So, this is what is systemic acquired resistance. Now, interaction of plants with non-pathogenic bacteria also induces resistance in the plant against pathogenic bacteria. So, this comes under induced systemic resistance in the plant. Many plants, although it is a hypothesis which we are planning to also test, that you have heard about endophytes, you have heard about mycorrhiza. Now, some of the bacteria, they live in symbiosis with the plant. So, now these organisms also first infect the plant. It is not only one give, there is a give and take process involved. It will be a mutual beneficial association always. So, imagine why would plant resist one and why would plant allow that particular organism to grow within. So, there has to be something beneficial from it, then only the plant. So, that is what we are also testing in our endophyte. So, in lab we also work on endophyte cultures. Now, these are microorganisms which live and reside within the plant tissues, and through years, through evolution, as the plant has grown, that symbiosis has developed. So, why only a particular species or set of microorganisms the plant is allowing it to grow and propagate throughout the plant, and it is not allowing the others. So, some of these, now any microbial, even to enter and live, reside within the plant, it will have to enter the plant. So, it is kind of an infection. So, think in those terms. Now, the plant is allowing that infection. In that process, although it is so, there is always a balance between the merits and the demerits. So, it is because of the mutual benefit which it considers to be much, it gives more priority over the demerit of infection, it is allowing that organism. But what other benefit the plant is getting is this induced systemic resistance. So, because of that, so for example, the signaling cascade where now jasmonic acid is involved or ethylene is involved. Now, salicylic acid is not involved. Now, jasmonic acid and ethylene are, ethylene is a growth hormone also, plays a role in developmental stages of the plant. Now, jasmonic acid is another molecule which plays a role in the signal cascade. Now, jasmonic acid and ethylene are the components which are involved in induced systemic resistance, which will increase the production of the secondary metabolites which are continuously being produced in the plant. So, there were some metabolites which are always present. no spatial or temporal induction of their biosynthesis, but there are phytoalexins which are produced only as a result of infection to the plant. So, this induced systemic resistance enhances the inherent capacity of the plant to protect itself. This form of systemic defense activation does not involve salicylic acid as the signaling molecule, as I said. It does not induce the accumulation of typical PR proteins. Now, certain defensive measures are immediately put in place by ISR. ISR was induced systemic resistance. So, this is actually innate. Now, other defense responses are initiated only after actual pathogen attack. So, that is induced, resulting in a faster and stronger response. So, everything comes together, all different modes of actions, defense are brought together. The advantage of this defensive strategy would be logically, without reading also you should be able to tell me. Why do you think the plant is having first line of defense, second line of defense, having different modes of actions?
Doesn't want to waste everything.
In the end, everything needs carbon and energy. And we know how difficult it is for the plant to get the carbon. We have now studied about photorespiration, photosynthesis, the loss the plant goes through. So, the plant has to judiciously use its energy to protect, and the priority is growth, always. All these come for survival, but ultimately the aim is to propagate, to grow. So, the plant has to prioritize all these together for the ultimate aim. So, therefore, it would not like to waste its carbon and energy all the time in this. So, it will have different stages. So, this is again a picture which can demonstrate to you systemic resistance, induced systemic resistance, where jasmonic acid and ethylene is involved. Again, it is through phloem, it spreads to other parts of the plant. So, they have shown in the root, if you can see, that there is a symbiosis between a bacteria. So, it is maybe forming a root nodule. So, because of this, the ethylene and your jasmonic acid production is induced, which further is involved in the signal cascade to enhance the secondary metabolism. So, you will always find that some plants are found to be very resistant, which will have very high antimicrobial activity. Not all plants have the similar antimicrobial activity. That is the very reason why some natural products are used as pesticides. Not all, like neem for example. So, there is a difference in the capability of each plant. So, from where is that coming? It is coming from all these. The different array of secondary metabolites which a plant would produce would depend on its systemic and acquired resistance. So, this is the summary of what we have learnt up till now. So, it is against the herbivore, the plant has to protect itself. Carnivore is involved or is involved further to protect HIPV. Any guesses? So, it is herbivore induced plant volatiles. So, secondary metabolic pathways, again to just to reiterate that there is a broad range of secondary metabolites, including classes like isoprenoids, complex isoprenoids. So, as I told you people earlier, that it begins with the primary metabolism, then major secondary metabolic pathways where MEP pathway, shikimate pathway, mevalonate pathway, these are in DX5 pathway, they may be involved for the backbone of those secondary metabolites. Then from these intermediates they can further come together, major classes can come together to form further more complex metabolites. Now, why do you think the plant is finding a need for combining further, branching it out, further branching it out?
Maybe to stabilize the compound.
Can be. What else?
It has a very basal structure, like conserved structure, and then just by changing the functional groups or adding some more, you can change the entire property of the metabolite for different actions.
Property in the sense bioactivity. If you see the chemistry, the mode of action of these complex metabolites, especially plant based products, they are very complex, large, molecules, organic compounds. Now their mode of action depends on a particular moiety or a functional group. It is not, now every moiety there has a different role to play. So it's kind of increasing the diversity of defense in the plant, or maybe to protect the plant itself. So that's the very reason possibly why the secondary metabolism is so widespread in plants. So secondary metabolites, synthesis and metabolism is in plant cells is by specialized proteins, represents the plant developmental process majorly, secondary metabolism. It is an expression of cell specialization. Now all these points are important to know. Why? So that when you are developing a plant based bioprocess for the production of a specific secondary metabolite, you will be able to know that what is needed in that bioprocess, whether a simple undifferentiated cell or homogeneous dispersed cell would work, or you need aggregation also to happen, or you need organogenesis to also happen for that particular metabolite. So when you know these aspects and you know your secondary metabolite and how is it and when is it and where is it getting produced in nature, you accordingly devise your bioprocess development. Like some particular secondary metabolites are not produced all the time. They are only found when a particular tissue root, they are only found in roots, or they are only found when the new leaf starts coming out, like in onion. I will be talking about those volatile components. So then if you know what is happening in nature, in order to produce that in vitro, you will have to create similar conditions. So expression of cell specialization, secondary metabolism is now triggered by the process of cell differentiation, because it is a higher order function. So it is generally associated with cell differentiation. The yields will be higher when the cell differentiates. Cell differentiation is a component of metabolic regulation in higher organism. Now it includes all process. When I say cell differentiation is from the cells to the tissues, then to the organs, and then to the entire organism. That's differentiation. It includes all processes which differentiate cell with the same genetic. So not all genetic information, although they are totipotent in nature, which means the genetic machinery is available, but not all genetic information is utilized all the time. So biosynthesis of secondary compounds is limited to therefore particular developmental stage most of the times, or specialized cells. Now where do they get accumulated? Now secondary metabolites, you know there are specific synthesis sites, storage sites, and generally synthesis is not the same as the storage site. Why? But they are getting synthesized, isn't it? They undergo.
So that it does not get.
The site where it produces might be far from the first line of defense. So yeah.
That can be a reason. Yes. What else? There is also the secondary metabolites. We know the plant will judiciously produce it only when it is needed. It will accelerate the machinery. So there is, and plant would not like to have it all the time because they may be toxic. So it will, there are turnover processes. So the metabolism, the rate of synthesis, and there is a rate of utilization. This also includes rate of transport if it needs to be stored somewhere. So there are turnover involved in secondary metabolites process. So therefore generally, and as I said, one of the logical reasons can also be to drive the reaction forward. So you would not like to, that's the reason why you will find sometimes add in resins for adsorption of the product, helps in increasing the rate of synthesis of the product. So that can also be a reason. So now they experience turnover processes with characteristics half-lives, some of these toxic secondary metabolites. Now degradation and synthesis often occurs simultaneously. the extent of accumulation depends upon cell synthetic capacity and capacity to metabolize the components. So, when we say metabolize the components, this also includes further modifying it for transport to the required places. Now, or modifying it for detoxification. Individual plant organ differs in their significance in this process. So, you will find that storage and synthesis, they occur in different sites. It is and sometimes the synthesis also is compartmentalized. Now, the concentration also varies according to geographical location, climate and soil. So, it is nature dependent. Now, spatial compartmentalization, synthesis is bound to particular organs, specific cell organelles or vesicles. So, synthesis need not be, like for example in camptothecin, there are reports in literature which say that if you try to extract camptothecin, you will find that you will be able to do it best in maximum yields from the bark. But when synthesis has been observed, there are literature reports which indicate that the synthesis happens in the root. So, which is an example that the site of synthesis may be completely different from the site of storage. So, example, intermediates in berberine synthesis travel back and forth between the cytoplasm and specific vesicles. This is for an example for spatial compartmentation of the secondary metabolite. Now, storage, there are different places and there are different forms in which the product can be stored. They can be stored on the surface in the form of trichomes, or even in the intercellular spaces, empty spaces, like for example azadirachtin, if you do, we were working on hairy roots. So, if you do a cross section of the hairy roots, you can do histochemical assay and find azadirachtin spots colored under the microscope, and they get stored in the intercellular spaces of the root matrix. Then if you can see trichomes are hair like glandular structures on the plant leaf or the stem. Then idioblasts, these are specialized plant cells which are meant for storage only. So, they are only meant for storage of either pigments or wax or oils or your secondary metabolites. Then glandular hairs, trichomes, cuticle, epidermis, there are different parts or tissues or specialized organelles where you will find these secondary metabolites will get preferentially stored. So, vacuole, it is a common site for short and long term storage. Vacuole inside a plant cell, if it wants to protect itself, then also it is stored in vacuole. Now, only two categories of secondary metabolites are stored in vacuoles. Toxic compounds are glucosidated for this purpose. So, when glucose moiety is added, it is one of the biotransformations for detoxification. Sometimes they do not store the final product, but rather it precursors. We have studied about it, where the enzymes for the final product are in the vicinity such that the intermediate compound, as soon as the cell ruptures, that comes in contact with the enzymes to give the final product, which is toxic metabolite for the pathogen. So, vacuole function as a trap for ions. Now, how does vacuole functions as a trap for ions? When we were studying about the different organelles and their functions, we studied about it. It is something to do with the tonoplast. The molecule is facilitated, it is generally not active process while entering. It enters, but going out is active or it is not allowed. How? We have studied this. How is it happening? Go back and read.
Because of the ion pumps on the membrane.
But I said it is a non-active process while coming in, generally. What facilitates that?
It says that interior of vacuole is acidic.
Right.
And ions can come inside.
Tonoplast is what? Is it no, there is no membrane across the vacuole? There is.
There is. That is
Membrane is made of?
proteins, lipids.
Hmm?
Lipids and proteins.
Cell membrane.
Proteins.
lipids.
Proteins.
So, what will allow easy passage? It has to be? Okay. Think.
Some portions.
I heard hydrophilic also. So, you need to think